I'm grateful for the Birnstiel award, and it is amazing to be here. And I would like to share a story from my PhD work on translation inhibition in bacterial CRISPR-Cas defense. I did most of the work in Vilnius University Life Sciences Center, and I was assisted by brilliant people working there. So bacteria have many different defense systems against different phages, which one of the systems that we are focusing on is CRISPR-Cas. During the infection, some bacteria in the population are able to hijack a small fragment of DNA and insert that fragment into the CRISPR region in the bacteria's genome. Later, this CRISPR region gives rise to CRISPR RNA molecules, which, together with Cas proteins, form effector complexes, which protect the bacteria against secondary infections. So the famous example is Cas9 protein, which was repurposed for genomic engineering, which finds specific foreign DNA in the cell, and then it cleaves it. My research revolved on a less famous, but way more interesting CRISPR-Cas system. So it is from type III CRISPR-Cas, and it is directed against RNA, not DNA. So once such a foreign transcript emerges in the cell, it is recognized by type III effector complex called CSM. It is a multi-subunit complex, and it has two enzymatic domains. One is CSM3, colored in green here, and the other is Cas10. CSM3 subunit forms the backbone of the complex, and it has RNA's activity against the target RNA. And the Cas10 protein is activated upon target binding. And also, it is worth mentioning that this complex is very well studied both from structural and functional standpoints. So during the defense, Cas10 protein cleaves the DNA that is being transcribed by its HD domain, and its POM domain converts ATP molecules into unique signaling molecules. They are called cyclic adenylates, and they in turn activate associated proteins. So most of these associated proteins have a CARF domain, which is required for binding these cyclic adenylates. The first protein described that is regulated in this fashion is called CSM6. And once it binds the cyclic adenylates, it shreds the total RNA in the cell. So this causes dormancy of the cell, but it allows the population to survive the infection. Computational analysis allowed researchers to classify the systems. So CSM6 and similar proteins belong to CARF1 group. And we were interested in another one, CARF7. CARF proteins in this group employ different effectors. And we were interested in potential RNases of REL-E family, and the proteins having these domains as effectors are colored in magenta here. So we selected 4 homologs for further characterization. So what is REL-E? Well, originally, REL-E comes from bacterial toxin antitoxin systems, where they act as toxins. And they normally are neutralized by antitoxin REL-B. But during certain stresses, such as nutrient deprivation or antibiotic presence, as early studies suggested, the REL-E becomes activated. Current view shifts towards another stressor, phages. But regardless of the mode of the activation, REL-E protein goes, when it is unleashed, it goes to the ribosomes, where it cleaves the mRNA being translated, causing the protein production shutdown in the cell. Now we solved a crystal structure of CARF-REL-E fusion protein. So the structure confirmed that it is a dimer. It is composed of N-terminal CARF domain, middle winged helix turn helix domain, and a C-terminal REL-E domain. When we incubated this protein with RNA, we didn't see any cleavage despite we added activators. But when we pre-assembled the same RNA with a ribosome and then added the protein, we see a prominent cleavage when we added cyclic tetraadenylates to the reaction mixture. And when we map the cleavage products, here, we see that CARF-REL-E protein cleaves the RNA in the A site, similarly to standalone REL-E toxins. And this activity allowed us to rebrand these CARF-REL-E fusion proteins as CRISPR-Cas associated mRNA interferases, or CAMI for the rest of this presentation. Knowing that they are activated by they require ribosomes, we also solved a cryo-EM structure of CAMI1 bound to the ribosome. As expected, we observed that CAMI1 squeezes in one of the rel-E domains into the A site to cleave the mRNA translated there. We also noticed these CAMI proteins change their conformation upon binding the activator. Most prominent change is in these H-stage domains because it reveals a surface for interaction with ribosomal protein called L12. Well, this puzzled us a little bit because L12 protein is not very often visible in ribosomal structures. So the reason behind this is that L12 protein comes from a very flexible structure inside the ribosomes called the ribosomal stalk. So it is present in every ribosomes, but it's not visible. The main function of ribosomal stalk is to form transient interactions with various translation factors. This allows to enhance the translation, and it is absolutely necessary for the protein production in the cell. CAMI1 is the first protein, well, at least to our knowledge, that is able to hijack this ribosomal stalk to enter the ribosome. So we perform biochemical and biophysical analysis of several amino acids from the H-stage site, and we also confirmed that this interaction is necessary for the mRNA cleavage by this protein. Coming back to CARV domains, we knew from other studies that some CARV domains are able to cleave their own activators. And we tested for this activity with CAMI1, and we also observed cleavage of the activator. Mass spectrometry analysis revealed a couple of products which result from a stepwise cleavage of the cyclic activator. We also found a mutant in which the ring nuclease activity was reduced. Finally, we wanted to test whether CAMI1 proteins are active in live bacteria. Since our laboratory of Wargehorse E. coli does not have any type III systems, we reconstructed them using plasmids that encode proteins, Cas proteins, CRISPR RNA, and the target for this complex. And this allows the production of cyclic adenylates in the cell. Then we transformed these bacteria with variants encoding CAMI1 protein and measured their growth. So wild type and ring nuclease activity mutant were unable to grow very well, but when we introduced another mutation in the mRNA interferase active center, we see that these bacteria grow as well as if no CAMI1 was expressed at all. We purified RNAs, total RNAs from these bacteria, and then searched for specific RNAs using radioactive probes. So in case of mRNAs for some of protein encoding genes, we saw that wild type and ring nuclease mutant was degrading them. But in case of ribosomal RNA, which is not translated, well, it was kept intact. Together, all this data shows that CAMI1 proteins are able to be toxic or to provide defense in other species, and mostly because of the conservation of the translation machinery. It could also explain their spread among bacteria. So putting all the pieces together, we provided a mechanism. So during the defense, cyclic adenylates are produced. They activate CAMI1 protein, which then changes its conformation and allows itself to be captured by ribosomal stalk. It enters the ribosome, cleaves the mRNA there. Due to this cleavage, these ribosomes get stalled on the truncated RNAs. The accumulation of such stalled ribosomes drives the protein production down in the cell. And this actually should give time for the cell to deal with the viral nucleic acids. And when the bacteria overcomes the infection, it should somehow come back from this translation inhibited state. So in a separate study, we observed that the RNA cleavage products leave the CSM complex. They deactivate the Cas10 protein, so it shuts down the production of cyclic adenylates. The leftover cyclic adenylates are degraded by the CARV domain, and the ribosomes are rescued using specific ribosome rescue pathways that are known from the literature. With this, I would like to thank people from Vilnius University Life Sciences Center that were part of the story. I thank my supervisor, Gintautas Tamulaitis, for assembling a team which makes doing science enjoyable. I also thank our collaborators from Leipzig University and our funding sources. And of course, I thank you for the attention.